Hello, mic check, check.
Is it on? Am I audible?
Yeah.
Probably too loud.
Okay, so let us get going. Good evening, everyone. As you know, of course, today we have this very special joint asset colloquium joint with TIFR Group for Gender Harmony. And this is in connection with the International Day for Science and Women and Girls. And this particular program was, of course, the day was on February 11th, but the nearest Friday where we can celebrate this was, of course, today. It is on 16th. As you know, we were planning to have Dr. Lalitambika from ISRO, who has kindly agreed to kind of talk today, but due to some unfortunate reasons, it couldn't happen. I'm telling all this because to say something about Vidita, how in the very, very last minute, she agreed to kind of give this talk and make the celebration happen uninterrupted. And it's not just that, even today she had some incident in the family, but still, in spite of all that, she comes and ready to give this particular colloquium. I really want to thank on behalf of ASET and all of us for agreeing to give this talk at such short notice. And your first slide already kind of made us to kind of glue to the screen. So without taking more of all of your time, of course, as I said, this is a joint colloquium, and I would like to, first want to thank Baswati, Sandhya, Kausika, and Vandana for making this program happen. And today, Vandana is going to formally introduce the speaker, not for you, of course, you all know Vidita, but there are also a large number of audience on the social media. So for benefit of everyone, so please welcome Vandana to introduce the speaker and start the program.
Satya. Good afternoon. As Satya already mentioned, this is a joint asset TCGH colloquium in commemoration of the International Day of Women and Girls in Science. Some of you are aware that this International Day of Women and Girls in Science was introduced by United Nations in 2016 with a hope to tackle some of the greatest challenges in various aspects of life, from improving health to combating climate change. All these aspects will rely on harnessing the talent, and it's essential to get more women working in these fields. Diversity in research expands the pool of talented researchers, bringing in fresh perspectives, talent, and creativity. This day is a reminder that women and girls play a critical role in science and technology communities, and that their participation should be strengthened. Although science, technology, engineering, and mathematics fields are widely regarded as critical to national economies, so far globally, no matter in what countries of their level of development, nobody has achieved gender equality in STEM, and we need to make further efforts towards that. More information about it, you can find on the United Nations webpage. And I actually was going to flash that here, but I really liked Vidita's first slide. So I thought that is what should form the background here. So this was started in 2016. So this is the 9th day. And theme for 2024 is Women in Science Leadership, a New Era for Sustainability. And we are very fortunate to have one of the reputed science leaders talk to us about her work today. As Satya mentioned, Vidita agreed to give this talk at a very short notice, and notwithstanding any other problems also. So her title of her talk is Life Experiences Shape the Brain, the Trajectory of the Lab. Vidita received her undergraduate training in life science and biochemistry at St. Xavier's College in Mumbai, so from this neighborhood. She obtained her doctoral degree in neuroscience at Yale University under the supervision of Professor Ronald Duman, and after postdoctoral fellowships in Karolinska Institute and Oxford University, she returned to TIFR as a faculty. This was in 2000. Currently, she is a senior professor at TIFR. She is a fellow of all 3 Indian science academies. She received the National Bioscientist Award in 2012, prestigious Shanti Swarup Bhatnagar Award in Medical Sciences in 2015, and the Infosys Prize for Life Sciences in 2022. In addition, she was awarded the Nature Award for Excellence in Mentorship in India in 2019. Her research group is interested in understanding the neurocircuitry of emotion, its modulation by life experience, and the alterations in emotional neurocircuitry that underlie complex psychiatric disorders like depression. She is committed to enhancing equity, diversity, and inclusion in academia. So let's welcome the one of our reputed science leaders, Vidita, for today.
Thanks, Vandana. I'm stepping in for a space scientist, and I'm not going to give you anything that leaves Earth terrain at all. So I'm just going to give you the heads up on that. I was also telling Satya that I have probably got the reputation of giving the maximum number of asset colloquia. So I don't usually ever say no to an asset colloquia, and this is just our own audience, so this is fine. So I'm going to talk about how life experiences shape the nervous system. And I'm going to tell you about life experiences I had as a PI when I joined the institute when I was a young kid, 29 years old, it was 2000, and how those have actually shaped our science. So for a Mumbaikar, coming back to the city was sort of critical. And when I was applying, this was my dream destination. So I came back here as a graduate student, came and gave talks as a graduate student to Veronica and Krishnan in the erstwhile molecular biology unit, which then became the Department of Biological Sciences. And the idea that you have this opportunity to work in this place on the tip of the most wonderful city of our country. I'm clearly not biased, but yes, the most wonderful city in our country, and that you get paid to just enjoy what you're doing is sort of surreal to imagine. And that I was actually coming back to a job here. I was just thrilled. I hadn't even asked, where is my lab? What is my lab? What is it going to look like? How much are you going to pay me? None of this was even a relevant thing. The fact that I had this job in this location, in this city, was itself pretty magical. I was fellow D when I got recruited. This position, I presume, now no longer exists. It has just disappeared into thin air, which is a good thing and nothing bad about that. So I come back and then I see my lab, right? This is my lab. Those of you who make it across to the other side would have heard about something called the Hutment lab. It is officially a hutment. It doesn't have a roof, right? So it is a hut structure. But I was, as I said, so excited at the thought that I have a job and someone's actually willing to take a risk on this 29-year-old who's come with some ideas in her head about what she wants to do, that I was just like very happy to be in this hut. 23 years later, my students don't want to leave this hut. So I guess there's something rather magical about that. We would like it if it didn't leak so much in the monsoon. That would be a nice quality, but overall we rather love our hut. Okay, so this is the Hutment lab, and this was actually where the National Center of Biological Sciences got started off. And this was Obaid Siddiqui's design. So there was a huge element of, oh my gosh, I am working in a place that Obaid designed. This is how he imagined a lab should be, and it was a hut. But somehow Obaid decided that a lab does not require a restroom. Why, I don't know. But he just eliminated the requirement of a restroom from the lab. I think presumably we were supposed to evaporate any additional, that was the requirement. So that was itself an interesting challenge. But anyway, this is March 2000, and we benefited in many ways from his design because yes, it's actually quite well-designed lab. And when I came back, I was interested in how life experiences shape brain circuits and behavior. And this was what I wanted to work on. So I'm actually going to tell you today a little bit about how obstacles and opportunities, people shaped the way we did science, the character of our lab, the personality of the lab, and then actually shaped our science. I'm going to tell you how this shaped this directly, and you'll see along the way how that ended up happening. So the first thing we wanted to do was populate our lab. And populating our lab meant human beings, but also meant these guys. We wouldn't do what we do without these really rather amazing creatures. So that's a rat. A rat is not just a large mouse. It's a different species. It's a rat. Anyway, these are our very, very cute rats. And no, this is a common question. Everybody seems to think they're just expanded, blown up mice, which they are not. They're a different species, right? So this is the OG crew in the lab. Initially, we got some money, and we were Y2K compliant. We got money from many different places. We got super excited that people were willing to take a risk on us and fund us. And then we had to go and get rats because there were no rats. So we went to, our first trip was actually to Hafkin Institute, where we went and bought rats. Now to bring them back was a thing. Like, how do we get the rats back from Hafkin? So I didn't want to put them in my car because that did not sound, and I had a really old Maruti 800, which I inherited from my parents at that time. So I put it in the dicky of the car. Then I said, these things are going to asphyxiate by the time I get back from wherever, Vadala, that area, and then come here. So I have to keep the dicky open. So me and my first graduate student, my first master student, tied up the dicky and kept it open enough, And then along the way, we have a taxi driver driving right next to us who realizes that, and he's looking at us most interestedly. And then after, at some point, we reach a signal and we stop. And he says, "Madam, aapko malum hai, aapki dicky mein chuhe hain." What did he think, that these things had jumped into the car and were riding with us? But it was just a reflection of some of the absurdities of how we got started. We had to get our second batch of rats from NCBS. And then we had to figure out which flight had pressurized cabins in which we could actually bring the rats. So that was actually a thing as well. So we do all this, and I'm thinking, it'll be really interesting to study how early adversity shapes these circuits, because we're interested in how neurocircuits in the brain talk to life experience and to drugs to actually modulate their function, and then eventually that emerges as changes in behavior. So we were interested in this, and this was not a novel idea, and we weren't the only people interested in this. People all over the world interested in it, starting from epidemiologists to psychologists to psychiatrists to neurobiologists. So here you see just a review article, which in a cartoon format describes how adverse childhood experiences shape the emergence of multiple outcomes across the entire lifespan. And this has been studied in psychology as well as in psychiatry for a long time. And when I say early adversity, what am I referring to? I'm referring to a variety and a spectrum of different adverse early experiences, which go from the spectrum of abuse to neglect to household dysfunction to poverty, which is also a major risk factor. And then these are, of course, intersectional in that you can have multiple factors at the same time. And based on the number of adverse childhood experiences you have, as these numbers increase, so does your risk for a variety of negative health outcomes, spanning from substance abuse to depression to anxiety, pretty much across the board. For every psychiatric disorder, early adversity is a common risk factor. So that's rare because there aren't that many common risk factors that hold true across the board. Okay, so we were interested in doing this, and we were interested in saying, how do we take something that happens in the environment of humans and model it in rodents? And obviously, we were not the only people thinking about this, and many people had been working on this. There are ways in which you can profile differences in the quality of maternal care, and this species, unlike this species, has a limited caregiver. It really has only the dam. That means it has only the mother, because that's the one who maintains the nest. On this side, it's a much more complex caregiver society. There are aunts, uncles, parents, grandparents. There could be, you could have children that are adopted. You have teachers. You have a structure of society that's taking care of the infant. In the case of the species we look at, this is actually just the dam in the cage in the animal house. So you can look at inherent differences in maternal care. And then there are perturbations like maternal separation, which I'm going to tell you about in a few minutes, which become ways via which you can model changes in the quality of this interaction, and then look at the consequences on the brain of the infant across the lifespan. So this was interesting, and now I'm going to tell you how else we populated the lab. So we get started, and within the first 6 months, I have students, etc., etc. And then somewhere along the way, I realized that I had not thought about one thing. What do I do after we take the brain out of the carcass? So we finish our experiment. Now we take out the brain. Once we've taken out the brain, there's still a carcass left, right? So we have to do something with the carcass. So I'm thinking to myself, this is a campus with physicists and chemists and mathematicians. I don't think we have a place where we can dispose of carcasses. We don't have an incinerator. So I figure my closest likelihood is the medical section. I'll go to the medical section. They must be taking samples from people. What do they do with all these samples? They must be incinerating them. So I asked them, I'd suffice it to say I did not get a response, and we left it at that. But I came back saying, oh, there's no incinerator. So now we have a bit of a problem. So then I said, okay, no, this is still a solvable problem. So we went to BMC. And we asked BMC, and there is a really good quality incinerator in Sivri. And, this is where all the medical waste from the entire city goes to be incinerated. So I asked the BMC guy that, we want to send our samples also to BMC. Is this a possibility? He said, no. So I said, why? He said, because it's not medical waste. I said, what do you mean it's not medical waste? He says, rats and mice are not medical waste. They are not medical waste in my book. Nobody said that a dead rat. He says, then he said to me, do you know the city of Mumbai? How many dead rats and mice there are? If we have to incinerate all of them, there will be no time to do anything else in the facility. I said, okay, fine. So then I was really stumped. So he said, but I have a solution for you. And he was obviously concerned about us. And I was making this trip with our first veterinarian who we had hired on a project position. And we asked him and he said, I have a solution. You go to this pet crematorium facility, okay? And you tell them you have animals that need to be cremated. So I said, okay, I mean, if this is the only option available, we went. And this was in Kandivali. So we went all the way to Kandivali and we asked the guy. That was a totally different experience. It wasn't BMC. They were very kind to us when we walked in. They made us sit down. They asked us if we wanted to drink water, etc., etc. And they said, what do you have to do? So we have to cremate these animals. So I said, yes, no problem. And we will give you the ashes. And then I thought, oh my God, this is a different league of a problem. Plus, independent of that, it was going to cost a lot of money to cremate each rat. And the scale of what we were doing with this was not even possible. I mean, yes. But so it was a failure. So we came back. And this is like, I'm describing this in a condensed version, but this was happening over months, okay? In the meantime, I can't stop graduate students from doing experiments. We already have money to do experiments. People have started, the animals are breeding, we have experiments going. So what to do? So we start populating the Hartman lab. We bought one -80 freezer, thinking anyway we need a -80 freezer. We will fill one shelf with the -80 freezer. We will put all the carcasses, pack them nicely and keep them and freeze them until we solve this problem. How long is it possibly going to take us? Maybe weeks, maybe a couple of months, maybe at the max 6 months, right? So this is what we did. And then we bought one. And then we bought one. And then we bought one. So exactly. So we had 4 -80 freezers. It was 4 years later. I could not figure out how to solve this problem. And by then, we were giving talks, we were publishing papers. How do I tell somebody that the place I'm running into a problem is not attracting interesting students who have very exciting questions to ask? No problem. I have great people in the lab. We have money. No problem. We've got money. We're not running out of ideas. We're not short on ideas. We have exciting things happening, but we don't know what to do with the carcasses. Many of that generation of physicists who I went to at that point in time asking for how do I solve this problem came up with very creative solutions. It's a good thing we have a lawn out there because somebody suggested, why don't you dig and bury? I wonder what would have happened to me if I, like a gopher, I had gone with my students digging holes across all of that beautiful lawn, burying animals every single place. I said, no, that's not an option. Somebody said, here's the sea. Have you seen the sea? Look at the scale of the Arabian Sea. I said, yeah, I know, I've seen it, but these things have paraformaldehyde in them. I can't put this into the sea. So 4 years later, 4 -80 freezers later, this was not a capital cost I had actually anticipated because it all filled up. Okay, everything was full. So at that point in time, this guy who was in the BMC had now got to know me and Yashwant, and I think Sheetal had just about joined. It was 2003. Just then he had joined. We had been going and he knew us by name. I knew him by name. I knew his children. I knew what standard they were in. I knew what they were doing. We used to sit. I had eaten every biscuit that was possible in his dabba, had drunk multiple glasses of tea, but it wasn't budging. He was refusing to accept these as biomedical waste. Finally, I said, listen, you will just save all of, he said, what about the other institutes? Other institutes have their own incinerators, or you have to go to the Sivri incinerator. So I said, you'll just save us. I know I'm the first generation. Many people will come after this, but you just make one exception. Note that rats and mice are also biomedical waste. One fine day out of the blue, he said, okay, I don't know what happened. Nothing other than maybe we looked particularly sad on one day when Sheetal and I went. Said, please solve this for us. We can't keep. I was going to think that I will retire in TIFR with 40 freezers because that's all I would have been doing, just populating all of these and filling them up. So at that point, he said, no, okay, fine. But I can't take you'll have to get the carcasses to Sivri by yourself. We will not send anything. We will not send any biomedical waste trucks. I said, okay, fine. We're rushing back to the lab. And we said, let's get one, not air-conditioned truck in those days, get a truck, empty these freezers, pack them and get them out of here fast. See, somebody's agreed, he may change his mind. So while he's agreed, let's get this done. So we do this. And of course, those of you who know the system know that there is something called an entry pass and an exit pass. How can we not know this, right? So there's an exit pass. So of course, we have to write an exit pass. So filled up all 3 forms of the exit pass. Obviously, this is non-returnable. It will come back only in ashes. There's nothing coming back. So I said, okay, fine. We go do this. So we write this. We write the exit pass. And the guy at the gate, as of, I think it was Mr. Kirkise, had a full-fledged conniption. One truck of saman is going out, which is not returnable. What are you sending out? Right? I said, so I'm trying to explain to him, no. So he said, can I open and see? I don't think you should open this. So we're having this conversation. Finally, he says, my Marathi is falling apart. I'm not, I'm a kid who grew up in the city, but I'm not a native Marathi speaker, but I'm giving my best shot at it. And I am telling him about how, this is so important. And now this guy's agreed and let's take it out there. So he said, Madam, मी म्हणतायत म्हणजे मी ऐकतोय, पण हा सगळ्यांचा entry pass कुठे? And that was the moment when I realized, okay, now my Marathi has completely fallen apart because I will have to explain biology at the level of procreation, ancestors, etc., etc. So along the way, and why am I telling you the story? Because at some point we realized that we can't fill everything and the space was getting really, really tight. So we had to let animals live long. So we just kept a bunch of animals that we did early stress on and we let them grow because it was easier to maintain them in cages than find room in freezers. So that opened a complete new angle that we had not looked at. And then we realized we stumbled on something that no one else had looked at either. Partly because in the US, you pay $5 a cage. So everybody's entire goal is get it done. Finish the experiment so that your bill does not become out of proportionally large, right? Here, thankfully, we don't have those kind of killer costs that are breaking our bank. So we let these animals go. We let them where we had to. We had no space. So there was not much choice on that front. And this became the genesis of a scientific idea. And that's what I'm going to tell you about now. So there are many models of early adversity. You can look at variations in maternal care. You can look at this model. It's a model that started in the 1950s by Seymour Levine, Paul Thotsky, and others, where you take the entire litter away from the mother for 3 hours a day from postnatal day 2 to postnatal day 14. So it's 12 days of daily separation for 3 hours. It's actually a very, very mild stress because in the wild, the dam would just forage for 3 hours to find food for her infant. So being separated for 3 hours is not a big deal, except that you're doing it in a laboratory situation in which she is not choosing to go out and forage. You have taken away her litter, moved her out, put her back and put the pups back. And that actually causes a deterioration of the quality of care that she gives. And what do I mean by quality of care? How do we even quantify it, right? So you can look at the number of nursing bouts that the dam has with her pups. The numbers of nursing bouts don't change. Their weight doesn't change. So they get the same amount of nursing, but the manner in which she nurses them and nurtures them changes. In that it goes into a passive position, laying on the side, where she's not doing licking and grooming and archback nursing while feeding them actively. So there is a qualitative difference in the nature of the nursing bout. So this was interesting. And there are many, many other models and some of these we use in the laboratory as well. But I'm going to tell you today about this model, which is what graduate students at that time got interested in. And normally everyone in the field had been looking at these animals till they became adults. And that was considered long-term. Because these are babies, we'll wait for them to become healthy adults, which is 2 months out. By 2 months, they have hit puberty, sexual maturity, they're full-fledged adults. We'll stop the experiment at 2 or 3 months. We just let them go. We let them go to 6 months, we let them go to 8 months, we let them go to 15 months and stumbled on really interesting things. Partly because I wish I could say we had thought this through so beautifully, but it just was a fluke discovery along the way. And then we got really interested. Okay, so there's many things that change with these models. There's enhanced anxiety-like behavior, enhanced despair-like behavior. I'll tell you how you study these two in these animals. You look at them using behavioral paradigms in which The animal does not have an ease of escape, and you look at how much of an escape attempt it makes. You look at its exploratory behavior in environments where there is potentially a strong safety/fear component. Cognition is their ability to learn a task, and I'll show you some cognitive tasks. The social interaction, their ability to have a fear response and then get rid of the fear response, which is equally important. Their ability to gate sensory information. This is what I want to tell you about, that we stumbled upon observations that indicated that these animals show strong accelerated aging and inflammation. And that's the story I'll tell you about today. Okay. And this is just summarizing that. This is what I'm going to tell you about. So if you walk away with just this much, you will have got the 2-minute blurb version of my talk, which is that early adversity is a risk factor that accelerates aging. That's pretty much everything I'm going to tell you. Now I'll dive into the data. Is it okay if I ask people to interrupt me if they have questions? Yeah, there's one already.
But since we decided to do something non-standard in the sense that after the initial adversity has happened,
right,
they become adults, you're letting them live longer. But what is happening during that period? I mean, you have to keep them entertained for
so I mean, they're in their cages, they are well fed, they have a nice air-conditioned suite, literally. They get distilled water, like double distilled water, etc. Plus, they're in good animal facility rearing. Are they in like real conditions? They also have social partners. I mean, they grow old with their social partners, but they don't have large burrow systems, which would be perhaps the most effective natural equivalent, because then you have a much more complex. So there's been a debate now about how impoverished the laboratory situation is. It's already somewhat impoverished, and they're healthy otherwise, quite healthy, but then they don't have access to what would be a much more complex societal structure. There are 4 animals or 3 animals per cage.
guessing that the initial stage you have some control, some different.
We have lots of controls that didn't go through maternal separation. And then there's a bunch that went through maternal separation for that 12-day window. And then we just let them be in the same environment. Yeah, and many times they're coming off of they're actually off of very identical genetic backgrounds. So otherwise we have a big variable that will come in from a genetic difference. So these are inbred rats. And so then we use multiple litters, because obviously one litter would not allow us to make any cows. Multiple litters. Yeah. But yes, they are left undisturbed across the lifespan until we do the experiments. Yeah. Yeah.
You mentioned when the dam was separated, caused her to be stressed.
It caused her behavior to change. When we tested if she was stressed, she was. So so you can measure their corticosterone levels, which is the stress response hormone, which is elevated in the serum of any individual when they are stressed. Because maternal behavior can change independent of stress. So these species have many, many litters. So their investment is proportional sometimes to the availability of the litter, whether they see there's a perturbation, because we use primiparis dams in that it is her first litter ever. And we never use animals that have had prior experience of raising multiple litters, because that becomes another variable. Because naturally, over time, if you let animals go, you will get a nice binomial distribution with high caregivers and low caregivers. That'll just happen if you let them raise multiple litters. These are all primiparis in that they've never had a litter before. So that everybody is just raising their first litter. Otherwise, we bring in massive variation from that component. In any case, even with primiparis dams, because there can be variability from one dam to another, we need large N's to be able to make sure that we're not worrying about one dam where even despite separating, she continues to be a good caregiver. We have those examples that show up. So we do know that, not from our work, but from other people's work, where you can just take the entire litter, adopt it across to another caregiver, and it's all gone. So you can get rid of a lot of those signatures just by cross-fostering. You also know that if you take the pups and you can stroke them and substitute for the caregiving, and that's Stefania Macari's work, which shows that, and it's a location in which you have to stroke. But if you do that, you also change a bunch of serum-related factors that also ameliorate the stress effects. So yeah. And across multiple models, different severity, you see this. So I'll just go and dive in. So Deepika, and this is a while ago. So people had been playing with this idea and letting these animals go for a length of time. And Deepika at some point, this was early 2006-07. At that point in time, she said, okay, you know what? I've let a bunch of animals really go far out and I'm going to look at their cognitive behavior. So I will explain this task. It's a novel object recognition memory task. It's a simple object recognition memory task. So what the animal has to do, here's the cohort. You have a bunch of animals that are either controlled or maternally separated. And then you wait, either check them at 2 months and have another cohort that you check at 15 months. So now you have cohorts that are being checked when they are young adults and cohorts that are being checked when they are middle-aged. And here's the paradigm. What you have to basically do is on the first day, you get them used to this box. Okay, there's a box in which they go, they spend some time in it. If they spend 3 days in it, the box is no longer novel. Now they have 2 objects, and the objects could be like a dumbbell weight, or it could be a glass containing sand in it, and it's just sitting there. And they're 2 identical objects. And you ask the animal to explore this. And of course, animals like new interesting things, especially because they sit in cages that are fairly boring otherwise. So there's at least a novelty element. So there's a strong desire to explore those novel objects. They explore it. And on the next day, you replace one of these now familiar objects with a novel object. So this animal now has to remember, hey, I saw this before, and this is what I need to now spend more time exploring. So you're asking for an object recognition memory, and this is just a simple discrimination learning to see, does it remember what it saw before? And then 5 days later, you give it another new object and the same familiar object, which it has now seen. This is the 3rd time it's seeing that familiar object. And you ask, do you remember this? And then a strong positive discrimination index would indicate that the animal can absolutely recognize the object and spends much more time with the novel rather than with the familiar. And it's a straightforward, easy task to do with rats. And at 2 months, you can see that it doesn't make any difference if you're controlled or you went through a history of early stress. You'll do equally well. So really, baseline cognitive tasks like this are not impacted by this history of early trauma. But when you wait till 15 months, something rather dramatic happens. First of all, they don't learn. And even after seeing the object 3 times like this, they're still not doing anywhere near as well as the controls. And that's a strong indicator that object recognition memory is already showing pretty strong decline. And this was interesting to us because this is an indicator that already in middle-aged life, you start seeing signatures of cognitive decline, which are normally associated with aging. So you should not be seeing this in a 15-month-old animal. It's like a 24-month-old animal that starts showing a deterioration of this kind of cognitive task. But we already begin to see this. So Deepika was interested in this. And one way we can actually look at all the genes that are showing transcriptional changes. So every cell makes proteins. All those proteins come from your genome because genome makes RNA, RNA makes protein. You can just say, agnostic to whichever RNA, I'm just going to take the whole hippocampus, which is a structure in the brain that is responsible for cognition, and I'm going to look at every gene transcript. So then I just get to bash the whole genome in a sense and ask what is regulated. And what's already obvious is that there's a barely any overlap between the genes that are dysregulated or altered at 2 months and the ones that are altered at 15 months. Many more genes start getting dysregulated as these animals age. And this is over and above what is there with the control. So controls, if you simply took controls which are 15 months and 2 months, you obviously see changes anyway because they're also aging. But in the early stress animals, there's a much larger scale of dysregulation. All right. So let me come back to this idea now, which is what's going on here? The question that I was being asked by Vijay, which is you did something here, then you just left them in the animal house forever and then looked at them here. How do we know what's actually happened in this window? And can we start exploring that and ask, when does this first show up? When does it start segregating to this degree? And what is actually going on in the brain? Why do we think this is important? There's emerging evidence from groups across the world now using humans who had either history of stress or had generational trauma. And this holds true for children and grandchildren born to Holocaust victims as well. So when you look for changes, even multiple generations out, where there has been a grandparent with a history of severe trauma, you start seeing signs of cellular aging. So all of us and our cells are, as they are dividing, are showing something called telomere attrition. And one way to think about this is all your genome, the ends of your chromosomes are capped with tails. Those tails start degrading over time. And as they degrade, they give you a sense of the cellular age of different cells in your body. If they degrade faster, despite you being whatever chronological age, it's indicative of cellular aging going much faster than chronological age. And that had already been suggested that, we start seeing this in individuals that have gone through severe early trauma. So that was interesting. And this is relatively recent, and there's been now a real upsurge of interest in what is actually happening after this that leads to all of these cellular aging-like phenotypes. So now I'm going to tell you what the next person who came in did. So this is Prateek. Prateek is an intermediate early career fellow from the Wellcome Trust DBT India Alliance, and he's been in my lab for the last 5 years with his own competitive grant funding. And what Prateek was interested in asking is, what's the hole in between these two, early life stress and accelerated aging? Can I study this in both the periphery and the brain? And I'll walk you through the data. Interrupt me if I say something that is confusing for whatever reason. All right. All of you who have taken a blood test, some of you at least might have looked at a value called C-reactive protein, CRP. Sound familiar? Yeah. It's an indication of the degree of inflammatory load your body is carrying. Okay. So here, now we are looking, and this is what Prateek is doing. He takes animals, P2 to P14, and at different time points, he looks at the serum and measures the CRP. And when you do that, please note that the y-axis is changing, which is natural, because as you age, inflammation anyway goes up, which is as you expect. But at each age, if you compare cohorts of 10 to 15 maternally separated animals versus 10 to 15 controls, what's apparent is at 2.5 months, nothing is happening. By 6.5 months, C-reactive protein is going up, and by 15 months, these animals show a profound inflammatory phenotype. We haven't done anything to them other than that initial perturbation, okay? So they've had the same environment, the same conditions, the same diet across their lifespan, but that history is enough to change the trajectory. So that was interesting. So when he saw that, he's like, okay, there's a indication that peripheral inflammation in the serum is up. Can we look at inflammatory markers? So you can look at what are called inflammatory cytokines. These are things produced by your cells that are helping you fight an infection, for example. They're also baseline produced in any case. And you can also look at those that are used to, so there are pro-inflammatory and anti-inflammatory cytokines. What he found was the following. So when you look at IL-6, so interleukin-6, interleukin-1 beta, interleukin-18, tumor necrosis factor, at just 6 months of age, when these animals are, adults, not much is really drastically changed. Okay, oxidized LDL is a measure of your LDL, which is carrying an oxidized signature suggestive of oxidative damage. So that's a way of asking, did that inflammation have a consequence, in a sense? And nothing drastic is happening. There are few parameters that are off, but nothing that indicates a very, very severe phenotype at 6 months. But when you start looking at middle-aged animals, something interesting happens. So these are males on the top, and these are females on the bottom. IL-6 is up, IL-1 beta is up, tumor necrosis factor is up. IL-10 and IL-4 are anti-inflammatory cytokines, and they are down. So you have the signature where pro-inflammatory is up, anti-inflammatory is down, CRP is up, suggestive of the fact that you have an overall inflammation profile. Okay. So if you look at this now, middle age 15 months, CRP up, oxidized LDL up, doesn't matter if you're looking at males, doesn't matter whether you're looking at females, insulin-like growth factor, IGF-1 down, mature BDNF, which is a trophic factor found in the serum and in the brain, vital for neurons and neuronal health, also down. So something is absolutely dysregulated because of that history of early stress. So now here we come back to this idea again. Yeah. So it'll vary. So these right now I'm showing you 10 to 12 to 15 animals, but we repeat these over and over and over again until we're absolutely certain, because we will see variability. Because these are, you have to just have large enough ends. Yeah. So, but 10 to 15 is a reasonable single capital N of 1, where we have both groups with 10 to 15 atoms. But then we'll do it multiple times to make sure that it's real. Yeah. Sometimes in parallel, sometimes in series. It's crazy because these so our experiments are sometimes 15 months long. It's little crazy, but the advantage of it is that, first of all, we're not going to get scooped because nobody else anywhere in the world is taking the time to do this, right? Partly the reason we started doing this is because we were sort of forced to do it. And now that we've stumbled on it, it's too interesting to let it go. But there are not too many groups. I mean, I can think of a couple of groups. Most people get their animals when they are aged. So they can buy them from a vendor and say, give me aged animals. Very few people are going to go in with an early developmental intervention and then wait 15 months for so because you go broke. Your grant will be wiped out just paying for the animals across their entire lifespan. So yes, now we've kind of hung ourselves on this in that our experiments have just become this long. So when Pratik and I are discussing an experiment, sometimes we have to say, do we really want to do this? Because it will take us 15 months to even get the answer. It's going to be that long, right? But some of these experiments are worth doing. It's taking us the time to do it, but we should do it. Somebody should do it, so maybe we should just do it. So here now, Pratik started looking at, I showed you peripheral inflammation. Peripheral inflammation is up. What's the central indicator of inflammation in the brain? Central cells in the brain that are your surveillance cells that figure out inflammation are called microglia. They're going to go chew up, if you have encephalitis, etc., viral infection, etc. That's what's going in and cleaning up stuff in the brain. They're phagocytic, actively phagocytic. They can come in and eat. And obviously, they are also too much of them, and activated microglia is a bit of a problem because there's limited territory in there and you can't have these cells absolutely going wild with that inflammatory activity. So we are looking now here at 6-month-old animals. And in 6-month-old animals, numbers of these cells in this part of the brain, the hippocampus, are not really changed. And these are just different divisions, some different parts of the hippocampus. Forget the names. It doesn't really matter. Different regions of the hippocampus. Nothing really going on at 6 months. But by the time we hit middle age, numbers are beginning to go up again. So something is absolutely going on. And that's just another image of the same showing you what the microglia look like. There are these red cells here. Okay. So now we have this situation. What is happening in the nervous system? Is the nervous system carrying signatures of strong inflammation like we see in the periphery? And what Pratik has found is that, I will just say, ABCD proteins, which are part of inflammatory complexes, doesn't matter what they're called. Things that we know come up when the brain sees inflammation, like NLRP3, ASC, NF-kappa B, not changed in adulthood. These are just Western blots to detect the proteins. And these are samples. Obviously, we look at much larger N's, but here I'm just giving you a sense that here in control animals, these proteins are much higher in maternally separated animals than they are in controls. And so this is what we look at. You can actually start measuring this as well, where NLRP3 starts going haywire, ASC starts going haywire, and NF-kappa B starts going haywire. And this we have to repeat multiple times, so smaller N's right now. Okay. So the question then becomes, did this history impact hippocampal neurodegeneration later in life? Because you end up with all these reactive microglia, bunch of inflammatory markers in this niche. If that's true, then proteins like the ones that are studied by Debashish and Sudipto, molecules like alpha-synuclein, become interesting because they are precursor markers telling you if the brain is in distress and whether it's giving you an indication of what is going to come, which is downstream neurodegeneration. And what we see is, I don't know what happened to that. Here's alpha-synuclein. And you can see that in maternally separated, this is very preliminary data, and we're going to be exploring this much more. It's interesting to look at, but it's indicative of molecules that we know form fibrils and end up creating sort of traffic jams within neurons. So that's actually a very interesting indicator. Okay. So early stress is accelerating aging. It's giving you some sort of change in inflammation in the periphery. Inflammation in the brain, what's happening to the powerhouse of the cells, which are mitochondria? What's going on with mitochondria, which we know are vital for neurons? By the way, neurons, 20% of or whatever you eat and all your energy production goes to your brain because that's what is actually eating most of it up. And so it's a very energy demanding and energy hungry organ. So in this, what do we see? Ignore the 6 months for now. There's some attempt, very interestingly, to compensate. So when it's red, it indicates that gene expression is actually higher. When it's green, it indicates gene expression is lower. At 15 months, when you look at the 15-month-old hippocampus, then multiple transcripts that are responsible for different components of the mitochondria all down. And inflammatory markers are up, strongly indicating that there's a dysregulation on a global scale in this circuit. I don't need to give you the details. I'll actually skip this part and get to the part that tells you what we think is going on. Forget how exactly the data has played out. What we are suggesting right now is in adulthood, the balance between making new mitochondria and degrading mitochondria is reasonably decent till about 6-8 months. And then as these maternally separated animals age, the amount of degradation goes up and the production goes down, giving you a dysregulation. I'm not showing you the data now because that's stuff that is still underway. This is just to give you a sense of the mitochondrial DNA content, which will tell you about mitochondrial mass, not change in adulthood, moving downwards as these animals age. But what is also obvious, and you will see over and over again, is there will be overlap. There are animals in which the phenotypes are worse. There are animals in which the phenotypes are not that bad. And I'll show you that as we go along. Yeah, show you this. So if this is true, then you should be able to take mitochondria out of the hippocampus and measure their ability to utilize oxygen, which is a critical measure by which they eventually give you energy. That's how it works, right? So cellular respiration, this is essentially the measure of that. And when you do that and you take out mitochondria from the brain of middle-aged animals that had a history of early stress, which is this red line, give them different substrates and look at the oxygen consumed. So this is the oxygen consumption rate. It's already obvious that for the same number of mitochondria, you do not use oxygen as effectively. So you have a cellular respiration is down. So OCR is clearly down. All right. Mitochondrial shape is also distorted from EM data. I'll just move into this part. So I want to end with like an interesting angle that is coming up. I didn't show you the data, but one of the things we had already shown before that is early life stress seems to affect the muscle and the periphery and the liver before it affects the brain. The brain is privileged. So if I look at a 6-month-old muscle, I can already tell that that muscle comes from animals that went through a history of early stress. But if I look at a 6-month-old brain, I can't. Because they look so similar at 6 months. The nervous system looks still pretty protected. So this was something Pratik noticed as well over time, that the signatures in the periphery are worse than the signatures in the brain. And the signatures in the brain come on slower than the signatures in the periphery. So when you do that, here's just showing you muscle. And this is now 6-month-old rats. So if you're looking at skeletal muscle and looking at mitochondria in this circuit, here's empty DNA, here's TFAM, it's already down. So things are already going haywire in the periphery before they go haywire in the brain. Okay. So then why? Why do you keep protecting the brain? And this is something that over and over again you see in evolution, which is that if you have nutritional deprivation, there's starvation, there's early life stress, etc., you try to privilege the brain and protect it as much as you possibly can for as long as you possibly can. And how do you do this? You do this through a structure called the blood-brain barrier, which doesn't allow stuff from the periphery to go straight into the brain, including inflammatory molecules, including many immune cells, including drugs. One of the big challenges is often getting the drug across into the brain, right? So the blood-brain barrier is going to select what it's going to let in and let out. So something is happening at some point where all that you saw in the muscle and the liver catches up with the brain. So Pratik got interested in asking, what does a blood-brain barrier look like? If I look at the blood-brain barrier, soon after early life stress, and I wait until these animals age and then look at the blood-brain barrier, what's going on? So that's what he decided to look at. This is looking at gene expression of blood-brain barrier components. These are multiple genes that produce the blood-brain barrier. In an adult, they're absolutely normal. There's really nothing changed with the blood-brain barrier at 6 months. In middle-aged animals, multiple components are already beginning to show compromise. But this is an indicator. It doesn't tell you that the blood-brain barrier is, you could still have gene expression changes and nothing to the blood-brain barrier. So what he decided was a very, very simple test, which is an Evans blue extravasation test. Evans blue, if you stick it into the tail vein, it's going to go everywhere in the body. And the blood-brain barrier, if it is intact, is not going to let Evans blue into the brain. So if you now take out brain tissue like the hippocampus and simply spectrophotometrically ask, is there Evans blue inside the brain? You should not really detect reasonable levels of it if your blood-brain barrier is working and intact and doing well. And that's what happens in adult animals. Whether you're control or maternally separated really makes no big difference. Evans blue is not inside the brain. And we know it obviously is inside the animal because you can see it in the periphery. But if you look at middle-aged animals, which are 15 months old and had a history of this kind of early life stress, something drastic has gone wrong with the blood-brain barrier in this period where they've actually largely been left undisturbed, which is that you have compromised the integrity of the blood-brain barrier. And after this, really, we think everything is downhill. We don't know how early this shows up. 15 months, how much earlier have you gone? A little earlier than 13. So we need to now work backwards in a sense to ask, where does this compromise first show up? Because we think that's going to be absolutely critical to understand the emergence of these issues that come up as these animals age. Okay. And that's something that is really interesting because a healthy blood-brain barrier will allow you to keep out all kinds of things. And an altered blood-brain barrier lets leukocytes in, allows all kinds of other things to come in. It allows leak of all kinds of other toxins, etc. So this becomes an issue. All right. So finally, Pratik has just started experiments where he's asking, can I at least start by cross-correlating the extent to which the cognitive changes arise with the signatures in the periphery, the extent of neuroinflammation with mitochondrial dysfunction, and just start the very first round of cross-correlations to ask, are the animals that show the worst cognitive scores the ones that have the most severe signatures? On which of these do they have very severe signatures? So here's just a repeat of what Deepika had shown, which is large cohorts of animals run on a novel object recognition task, short-term, long-term in males and females combined. And what's already apparent is both short-term and long-term discrimination of object memory is compromised if you had a history of maternal separation. What's also obvious is there are aged animals who never went through maternal separation who are also impaired, and then there are animals that are unimpaired, but the distribution is different in this cohort. So there's something going on in the maternally separated cohort. And when you start looking at cross-correlation analysis, you're now looking at C-reactive protein, IL-1 beta, tumor necrosis factor, and an anti-inflammatory cytokine. So the higher the CRP, the lower the discrimination index. So the worse the inflammatory signature, the worse the performance on the task. But what was interesting to us is when we combine these animals, both the maternal separated cohort and the control cohort, in the control cohort, yet none of these have shown up as strong correlations. So there's something going on here where the correlations are strong in the maternal separated cohort. We predict that eventually, as the controls get even older, that these will start getting more correlated, but we haven't done that experiment, so this is conjecture at this point. But this is something that will be interesting to explore. And when you put them next to each other, this is what you see. Here's CRP. Here's CRP in the maternally separated cohort versus CRP in the control cohort. IL-1 beta, and you can just see the discrimination indices versus the concentration of the particular interleukin. And this is an anti-inflammatory interleukin, which goes in the exact opposite direction. So when we received, somewhere along the way, we received money from the Padmavati Venkateshwar. I do have 2 more minutes to wrap up. Yeah, so I'm nearly done. So to say, can you reverse this? What is the point of studying all this if you can't go in and change it? So obviously, everybody wanted to ask, can you make this go away? And naturally, as neuropsychopharmacologists, we were interested in using drugs. But they were least interested in our using drugs because they're not something that you can potentially translate easily. So the Padmavati Venkateshwara Foundation gave us a really generous grant for 3 years. And they said, please do experiments with things that are OTC, over the counter, which means we could scale this as a public health intervention if this becomes of relevance. So then we were scratching our heads. We had to find something OTC, we have to find something cheap, we have to find something that we can give our animals for many months because obviously we also have to be able to afford doing that experiment. So along the way, collaborators, Pratik, I, our grant committee, everyone had many different suggestions. And what we went with was nicotinamide, which is basically vitamin B3. Okay. It's commercially available. It's the easiest thing to get. It's cheap. We can bulk provide it. The reason we picked it is it's a precursor in a pathway that is vital for bioenergetics. And since we see dysfunction of mitochondria as a component of the inflammatory signatures, we wanted to see what this molecule would do, besides which in India, we have B complex deficiencies on colossal scale, right? So if you overlay early stress and then this nutritional deprivation deficiency, what happens? So we were wanting to ask, can we supplement with this and see something change? Maybe, maybe not. So yeah, these are the experiments Pratik has done. At 8 months, he's either got controls and maternally separated animals drinking water, regular drinking water, or he's giving them nicotinamide in the drinking water from 8 months to 13 months, after which they run through the behavioral task and then we look at the brain. We haven't yet finished looking at the brain, when these experiments are still underway, but I'll just give you a sense of what's actually happening. Our goal is to look at discrimination learning, look at the brain, look at inflammation and aging associated markers in the periphery, look at the blood-brain barrier integrity, which I think is perhaps our most dramatic finding, that the blood-brain barrier is so strongly compromised, and then look at obviously cross correlations as well. And this is just one, capital N of 1. So jury's out yet, but I want to show you some tantalizing data that's really interesting. We need to confirm it. We need to look at it better. We need to really do many, many more experiments. So here are control animals. That's their discrimination index when they are 13 months of age. Here, maternally separated animals that are drinking regular water. So both of these are on regular water. You can see that the maternal separation phenotype is a robust phenotype. No matter what the variation, there is always a decline. And we've seen this now in so many cohorts over the years. These are control animals drinking nicotinamide, and these are maternally separated animals drinking nicotinamide, and something is going on. Enough for us to feel like we need to go look at this a little bit more closely, check it a little bit more closely. Okay? So we think it improves cognitive behavior, but like I said, until we do this again, these are just the cohorts that we have. These are the ends that we have so far. We need to increase this. Skip this part. All right. So at this stage, we are interested in how you might have this. You have this long window after early life stress. And in fact, all these changes don't come up till 6 to 8 to 10 to 15 months. Can we not then potentially come in and intervene with a variety of different interventions? I'm going to wrap up by acknowledging all the people who worked on this. Pratik has led this entire project, and it's wonderful to have him as a collaborator and colleague. He's been ably assisted by Astha. We couldn't do these experiments without multiple other people in the team, including from my lab, as well as people from Ullas's lab. But none of this is possible without Doc hanging in there and letting us age our animals for 15 months and 16 months, and never asking us, saying that we're crunched for space. I have the rare privilege of collaborating with my dad, who has been wonderful to talk to about this project. Ullas is our collaborator on this project as well. And Carmen Sandy at the EPFL is again one of our major collaborators on the study. Our funding comes from TIFR DAE, the Padmavati Venkateshwara Foundation, a Fulbright Fellowship that I recently had, an India Alliance Wellcome Trust, and DBT Fellowship to Pratik. And I can stop and take questions.
Always one. I didn't even summarize in one line, and there are so many questions. So I was just about to say, it's always great pleasure listening to Vidita, and one never gets tired of. So that's why many asset colloquia. So there are, I think I will refrain from saying anything else, but let me start.
And all this is still unpublished. Just giving you a sense that I just showed you all the raw data that's come out of the lab last 4 or 5 years.
Yeah, we'll come to, but okay, so let us.
I came late, but it was wonderful. And I just wanted to ask this quick question. Look, Vidita, this is a very slow process.
Oh.
So, and that's why the challenge in the experiments. But if you were to think of chemical ends of where the descriptor lies in terms of the molecular processes, you identified something morphological already, that the blood-brain barrier is compromised. However, 13 months to compromise the blood-brain barrier seems like a reaction that is going on, or a process that is going on,
Of course.
which is absolutely a rare event in most of the lifespan. But it accumulates
Yep.
somehow to sort of have a confluence of multiple kinetic pathways. So can you point a finger to what those early events, the rare events in the early part of the aging process allows for? And if you can give a different stress to the
Mhm.
same animal,
Mhm.
or maybe repeated stress, can you just amplify that process and look at that chemical process much
So
more easily?
adult stress doesn't do this. It'd be really nice if it did. I mean, it would not be really nice, but it would make our life easier. It's good that adult stress doesn't do this. If you come in with stressors in adulthood, short-lived, even very severe, it messes things up in the short term. But there is really rapid recovery. The system is resilient to adult onset stress. And that's what the system is beautifully designed to do. There's something about the early window that's called a stress hypo-responsive period. That window in which we are doing this perturbation is when you're setting the tone for the rest of your life. Because the stress hormone axis is developing in that exact window. Okay. And it's called the HPA axis, the hypothalamus, pituitary, and adrenal axis, which produces cortisol in us and corticosterone in the rodents, which is essentially a cholesterol-derived small steroid hormone. But that hormone does everything in your body in terms of driving a stress response. Aging is a stress, but it's a cumulative, slow stress. People have gone in with adult stressors in animals that had a history of maternal separation. So that's the second hit. On the second hit, you don't see an aging-like change, but you see lots of things go haywire if you come in with a second stress early. But the system has a great way of buffering, and which is why the natural process of aging, it's not so accelerated that at 5 months, suddenly you're going to see an aged nervous system. Not at all. This stuff is slow, and it is attrition, and their lifespan is about 2.5 years. So we are picking this up but this we know from all the epidemiological work, that the system buffers until it can't buffer anymore, and then it crashes steeply. So in a sense, if we do pick something which moves it shorter, we will be looking at a different problem. We'll be looking at the problem of how does that history change the way you buffer severe adult stressors, not the slow cumulative consequence of the natural process of age. So yeah, I mean, now we've sort of signed on to this. So at least we have to finish this. I don't know if any of us is desperate to do this all over again. It is quite hard to, do the experiment and wait 15 months for things to finish. But now I think we've stumbled on something interesting enough that we will have to take it to some. I mean, the system is buffering across all reproductive healthy states. Essentially, if you look at it from an evolutionary lens, it doesn't really matter after you're reproductively senescent what really happens to you on species scale, because you've buffered and adapted in the early window. So there's this whole idea that you use all your resources to buffer early as best as you can. And when you do that, you will pay a cost. So there isn't a free lunch. Eventually you pay a cost, but that cost is cumulative and only shows up later. So on a species scale, it's still adaptive. It's maladaptive on an individual scale.
Hello. Yeah. So I think one of the interesting results was that the signatures of early stress are visible in the muscles before they are in the brain, right? So my question is that how much of this research has already been mapped to humans, and could we do something like measure in, say, someone's teenage, you study their muscle cells and detect those signatures of early stress in life, and probably, treat it with therapy, or let's say the drugs that you were talking about, and see the aging slow down or something like that?
People have done experiments where they've looked at telomere length in lots of peripheral cells in individuals that went through early trauma, either themselves or their parents went through early trauma, or their grandparents went through early trauma. So there are fair number of studies that indicate that if you look at a cellular measure of aging, it's speeded up in the periphery, because that's all you can measure in humans. So what you can get is PBMCs from so these are peripheral blood mononuclear cells, and you can look at the genome there, and you can ask, how quickly are the cells aging? And what you can see is they're aging faster. But you don't have, the thing is, you can't get muscle biopsy. You can't get liver punches. So these become difficult because somebody will have to go, be willing to allow you to take a little bit of their skeletal muscle out. No, it's not happening. Right? So blood you can get, serum you can get, all of that you can get, and that has been profiled. But there's variation because obviously it depends on the kind of early trauma. And you have to remember that in humans, you have massive genetic variation that will overlay on all of this. Genetic heterogeneity is a huge factor. Individuals do not respond to stress identically. We are looking at an inbred population where the genetic variation is dampened down, so we can now really look at an environment effect more easily. In humans, there's a gene-environment interaction. Your ends will have to go into thousands to eventually find something that is robust enough. But there's still, despite that, the telomere attrition data is strong. And there's enough reason to believe that it's intergenerational. That even if you have a grandparent that was in the Holocaust, that's the data, you will show telomere attrition in your cells 2 generations out. It's probably because you epigenetically prime the stress response pathways fundamentally differently, and then some of those signatures carry over to the next generation.
So essentially, your gene gets modified because of...
Not the gene. How the gene expresses gets modified by using... So think about it as chemical tags around your genes that determine how those genes will produce their eventual. So that's what's getting modified. Genetic modification will be mutation, essentially. Yeah. That doesn't happen.
Thank you. Great talk, by the way.
Oh, go there. Just a second. So we take one question from Vidita.
Yeah.
From the Zoom.
Can you hear me, please?
Yes, yes.
Yeah, okay. So actually, part of the question was already asked by gentleman before, but I would like to still ask, are there any non-invasive methods in humans that you could use to look at some of these transformations in the brain for individuals who have been stressed in their early life?
Not in the brain, but in the periphery, there's an idea that we've been talking about for a while. Microglia in the brain are most closely related to macrophages in the periphery. So macrophages we can get because it's from blood. And macrophages go from what is called an M1 to M2 transition. Essentially, macrophages become very strongly inflammatory, and how quickly they become inflammatory is an indication of the degree of inflammatory load or inflammatory susceptibility in that individual. That's a way of asking whether early history changes the priming of these cells to make them much more pro-inflammatory. That's something we can do. Unfortunately, in the brain, I mean, you can image, but that will not give you, we are not seeing volumetric changes. What we can look at and what could be looked at is the blood-brain barrier, because that is something one could actually look at. I mean, there are things like gadolinium that people use. Yeah. So people use that to ask about permeability of the blood-brain barrier. But that is unlikely to be a study that anyone will ever do with an early stress history. I mean, unless there is a strong reason to go in and there's a tumor or something of that nature, nobody is going to do those things in humans. So I think we are still, for the moment, going to have to study this better in rodents and potentially the field in primates as well, and use the periphery in humans to look at the periphery. Well,
thank you. Thank you.
Yeah, then one question. Yeah, please go ahead.
Wonderful talk. So my question was, so you talked about a few sort of physiological experiments, and then there was this one behavioral aspect of what's happening to cognitive this thing. Are you planning to, or is there any thing about looking at how, since you have long-term access to these animals, how does early life stress affect their social behaviors, because they're also together while they're in the cage and so on? And then one thing was, you had mentioned exercise as trying to see if you can mitigate whatever has happened. So did that show up?
Yeah. So we've not looked at social behavior at that age. We've looked at social behavior in animals when they were about 6 months. And actually, social behavior is not drastically changed in maternally separated animals when they're adults. social play behavior when their adolescence deteriorates. That means. So the precursor to social behavior in adulthood is deteriorated. They show disrupted fear responses. That's when they are young. They show disrupted approach and tasks where they have to go out into something which might not be the safest space. They show changes in those kinds of behaviors. That has been extensively reported, but that tends to be early. This is all in the window when they are 2, 3, 4, 6 months. The cognitive changes actually are interesting because unless it's a very severe change, very severe early stress, the cognitive changes actually seem to be an attempt to buffer. So you see these animals do better on stress-associated learning tasks in the short term, and they outperform controls, which was a paper we published a while back where it's biphasic. So they do better on tasks and then it's all down. So it's almost as if the system is using this to buffer the initial period and then there's a cost later. Exercise, actually, Astha is the one who works extensively with exercise in the lab, but we have not had the courage to put our animals on running wheels and watch them across the lifespan. We've done a lot of exercise stuff while the animals are young, and it's a very powerful intervention in terms of what it does to the nervous system and what it does to inflammatory signatures. But I think at some point we are just reaching the point where we are not able to sustain these very, very long-term experiments. So I think graduate students certainly should not be signing up for this on a regular basis with very, very long experiments. So no, I don't think we'll go down the road of exercise. I think we will go down the road of really exploring vitamin B3 much more closely.
Yeah. Thank you.
Okay, let us take that as the last. Rest, of course,
yeah, we can chat outside.
So there was some data about mitochondrial dysfunction, reduction in mitochondrial numbers, and other markers of dysregulation. So is there some plan to look at how mitochondrial morphology is being affected, and is that a mechanism of like this reduction in efficiency? Maybe
we can't
modeling
proteins. We
proteins.
can't do mitochondrial morphology in a dynamic manner, which is the best way to study morphology, which is to watch fission fusion. Partly because we have to look within the nervous system, and then we have a snapshot image rather than the dynamics of mitochondria. We're going down the road of doing EM, which will allow us to look at cristae architecture, look at shape, volume, etc., etc. But that's again a snapshot end state. It doesn't give you a sense of how, So our compromise on that has been to look at function and say, okay, we don't know anything about the dynamics because this is just too difficult for us to do in this cohort, but we can tell you about function categorically. And we know function is clearly compromised, and it's compromised because it's normalized to the mitochondria that you load. It's efficiency that is compromised. It's not just that you have less mitochondria, so you have less function. Per mitochondria, you have deteriorated function. So that's a strong indicator of compromise. But I don't think we'll be able to do the experiment. I mean, it would be lovely, but I don't know how we can do this in this system. We would need an in vitro system in which we can image them.
Okay.
Early stress, is it the only reason to change the telomere length or anything? If not,
of course, I mean, there's DNA damage itself. There are a variety of other ways you can cause telomere attrition. Other toxins will do it, etc. But this is amongst the mildest sort of stressors you can imagine that could potentially do this. And of course, it does it over a while. It doesn't do it immediately.